Hello, students. I'm back with my chapter. I hope you all are doing well and utilizing this lockdown and pandemic time as a quality time with your parents. This is a very nice time in this which you can discover yourself. You can work on your hobbies, whatever you like, what you want to be. Just give finishing to your hobbies, children. Apart from that, do concentrate on your studies, as studies are one of the most important part for your life till you get to your jobs. Now, coming back to our syllabus, today we'll be dealing with cytoplasm and its structure, cell organelles, and various structures that exist inside the cell organelle or the cell. As we all know that these cells perform distinct and a specific function. So we'll look to them one by one. Coming to the definition of cell organelles. Now basically we have studied about cell. Cell is having cytoplasm. In that cytoplasm, different parts are present and they perform different and valuable functions. These parts are often called as cell organelles. Coming to the very first part, that is Golgi apparatus. Now, Golgi apparatus consists of a set of membrane-bounded, fluid-filled vesicles, vacuoles, and flattened cisterns. Now, cisterns are the closed sacs. These are usually arranged in a parallel form to each other. There are two phases, cis phase and trans phase. Both are responsible for the working of synthesis of different things. Apart from that, incoming transport vesicles are there. Then newly formed vesicles and secretory vesicles are there. The gap between each system, in between the cistern, is known as lumen. That means a narrow gap. Now, functions. The main function of Golgi apparatus is to store and modify packaging and dispatching of the substance. It involves the synthesis of cell wall, plasma membrane and lysosomes. सेल वॉल, लाइसोसोम और प्लाज्मा मेम्ब्रेन के सिंथेसिस में सबसे बड़ा रोल होता है गोल्जी बॉडीज या गोल्जी एपरेटस का. Next is ER, that is endoplasmic reticulum. Now, endoplasmic reticulum is a membranous network of tube-like structure which extends from nuclear membrane to plasma membrane. न्यूक्लियर मेम्ब्रेन से प्लाज्मा मेम्ब्रेन के बीच में होता है. It is absent in prokaryotes and mature RBCs of mammals. That means us. We are having mature RBCs, so their ER will be absent. Now, endoplasmic reticulum is of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. The biggest difference comes in the presence of ribosomes on them. When endoplasmic reticulum is having ribosomes, it is or granular structure, that will be rough endoplasmic reticulum, and without ribosomes will be and smooth endoplasmic reticulum. There is the big difference. Now, if we talk about rough endoplasmic reticulum, so rough, that is granular endoplasmic reticulum, it helps in the synthesis of protein, that is formation of protein, and smooth endoplasmic reticulum, that P diagram, agranular endoplasmic reticulum, it helps in the secretion of the lipids. Lipids are very important for our body, as well as proteins are very important for our body. Function of ER. It gives internal support to cell, helps in the transport of various substances from nuclear membrane to plasma membrane, and vice versa, that is from plasma membrane to nuclear membrane. Rough endoplasmic reticulum help in the synthesis and transportation of protein, while smooth endoplasmic reticulum help in synthesis and transportation of lipids. This is the major function of ER. Coming to ribosomes. Now, they are extremely small, dense and spherical bodies which occur in a free-form matrix known as cytosol, or sometimes remain attached to endoplasmic reticulum. They are made up of RNA and protein, ribonucleic acid, single-stranded nucleic acid and protein. This is the structure of ribosome. It is made up of two units. One is a large unit which is in purple color shown to you, and another one is a small subunit. They both attach together, large and small unit, and form ribosome. Now, location of ribosome is given. See, you can have a look that they are freely present, and one more is there which are present on the endoplasmic reticulum that I have already shown in RER example. Now function. The major function of ribosome is for the synthesis of protein. Coming to the one of the most important part for our energy, that is mitochondria. Now mitochondria are basically small rod-shaped organelles that contain their own DNA and ribosomes. It is a double membrane structure. You can have a look. Double membrane is there. Then it is having DNA which is given in red color. Apart from that, it is having its own ribosome. Those ball-like structure in orange color are given. It is having cysteine. Cysteine is the location where ATP molecules attach themselves. Whenever we need energy, that ATP adenosine triphosphate breaks into adenosine diphosphate and phosphate ions and provide us the energy. Matrix is there which helps in the nourishment, and intermembrane space is there between the two membrane spaces there. Now, if we talk about DNA in deep, so they are small rod-like organelles. They contain their own DNA and ribosome. They are absent in bacteria and RBCs of our mammals. They are even absent, RBCs also does not have RER. You need to remember whatever thing is present in us and whatever thing is absent in our cells. Function. They are the site for cellular respiration and provide energy for the vital activities of all the living cells. They store energy and release them during the reaction. In the form of ATP, they are stored. That is why mitochondria is known as powerhouse of the cell. Without mitochondrial activity, we does not have any energy. Next is centrosomes or centrioles. Now, centrioles are formed only in eukaryotic animal cell, that is us. It is not bounded by any membrane but consists of centrioles, which helps in the formation functions. Now, centrioles are hollow cylinder-like structure arranged at right angle to each other and made up of microtubules. You can have a look. Small cylindrical structures are there. Total they are 13 in number. They are arranged in a microtubular formation. This is the location of centrosome in the cell. Functions. Now, centrioles helps in cell division and also help in the formation of cilia and flagella. Now, cell division means formation of parent cell, then from that parent cell, formation of two daughter cells. Cilia and flagella are the locomotory organs which are present in the prokaryotic cells, bacteria and other. They have flagella, a single form for locomotion. Cilia are small hair-like projections which are many in number. This is another a diagrammatic representation of centrosomes, how they are present, where they are located, their structure of the centrosome, and then from centrioles to microtubule formation. Coming to plastids. These are present mostly in the plants and absent in the animals. We all know they are usually spherical or discoid in shape and double membrane bounded organelle. Now it is double membrane bounded. You can have a look at its membrane. Outer and inner membranes are there. In between those two membranes, intermembrane space is there. Then inside that inner membrane, stroma, that is aqueous fluid, is there. In that fluid, particular structure is there, that is a granum and a stack of thylakoid. A thylakoid is a group of granas, and grana is a cone-like structure in which glycolysis carries out, that is formation, that is the conversion of carbon dioxide and water to energy, that is glucose and oxygen takes place. Now, plastids. There are majorly three types of plastids, that is chromoplast, chloroplast and leucoplast. These three are subdivided as amyloplast, eleoplast and proteinoplast. Now, if we go from left to right, then chromoplast. Chromoplasts are colorful plastids they does not have green color, but rest of the red color, yellow color is there. So if you see that money plant leaves and maple leaves, they are having yellow color, then orange color. This is due to the presence of chromoplast. Then comes the chloroplast. You can have a look. It is green in color, which consists of green pigment called chlorophyll and helps in the process of photosynthesis. Then comes the leucoplast. Now these are colorless plastids. They does not give any typical color, but they help in giving white color to the cell. So these are the three main ones: chromoplast, chloroplast and leucoplast. It attack Etioplast or theoplast is there, which is a combination of proplastid and etioplast. Normally they are formed and they are help in the discoloration of these cells. Then amyloplast, eleoplast and proteinoplast are the parts of leucoplast. Function of the plastid. Now plastid consists of chlorophyll, so you know that chlorophyll traps solar energy and utilize it for the manufacture of the food. Next, chromoplasts are the plant important pa- uh, impart various color to flowers to attract insect for pollination. Red color, yellow color, blue color, all these color are there on the petals of the leaf. Petals of the flowers. This is due to the presence of chromoplast, which helps in the attraction of the insects for pollination. Comes the leucoplast. It helps in the storage of food and formation of starch, protein, and fat for the plant body. Then comes lysosomes. Now lysosomes are the small lysosomes are the small spherical sac-like structure which contains several digestive enzymes enclosed in a membrane. They are found in eukaryotic cells, mostly in animals. Now that is the location of lysosome. If we see the structure, that is a diagrammatic structure which is showing it is a single membrane bounded structure in which protein and enzymes are present in it. Now, lysosome is also known as a suicidal bag because it helps in the digestion of foreign substance and worn out cell organelles. Whenever any cell part dies, then this lysosome engulfs that particular part. They provide protection against bacteria and viruses. Any bacteria or virus invades in our body, then these lysosomes absorb them and then they try to burst out in cleaning them. They help in cleaning the cells. Then, during the disturbance in cellular metabolism, like when cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as suicidal bags. Now, what exactly happened? Suppose a cell gets damaged, some any kind of activity take place inside our body जिसकी वजह से हमारा सेल जो होता है वो डैमेज हो जाता है. तो ऐसे में क्या होता है, जो लाइसोसोम होता है वो अपने आप को बर्स्ट कर लेता है और एंजाइम अपने आप को खुद के सेल को डाइजेस्ट कर देता है, इसलिए उसको सुसाइडल बैग कहा जाता है. इन दिस वे what happened, the other cells does not get any harm or damage because a single cell which was damaged is being busted out. Coming to vacuoles, they are liquid solid filled membranous cell organelles. If we talk about vacuoles, then in plants vacuoles are large and permanent. In animals vacuoles are small and temporary. Normally vacuoles help in giving lightness to the particular part. Suppose plant leaves are not having big vacuoles, then they will not be able to move here and there at the time of wind blowing. And similarly, if animal cells are having big vacuoles, then we will not be able to stand properly erectly. Now if we talk about the mature plant cell, then mature plant cell occupies 90% space of cell volume. Due to this size, other organelles including nucleus shifts towards the plasma membrane in a plant body. Now if we talk about, then they help to maintain the osmotic pressure in the cell. They provide turgidity and rigidity to the plant cell. This is the internal structure of the vacuole. This is central vacuole, main vacuole which bigger in size, which is present in a plant leaf. It is having tonoplast and cell sap. Now another one is the more bigger elaborated diagram in which location of the vacuole is given in a plant cell. Peroxisomes. Now they are small spherical organelles which contain powerful oxidative enzymes. They are bounded by a single membrane. They are found in kidneys and livers of the living organisms. You can have a look at the diagram. Little more deep you have to go left hand side, down side you can have a look. In between lysosomes and plasma membrane location of peroxisomes is given. I know it will not be extremely clear to you, but this much will be sufficient for your class 9th. If we talk about function of the peroxisomes, then they are specialized to carry out some oxidative reactions such as detoxification or removal of toxic substances from the cell. These are the cell cycles which take place. Now it is quite typical one, but just a rough one I am showing you. This is type of reaction of detoxification which take place in the peroxisome, the smaller structure, and forming a big reactions and a cyclic changes. Then coming to difference between plant cell and animal cell. Now the have as you look at the diagram you can go through it that plant is having cell wall and cell membrane while cell is having only cell membrane. Cell wall is the rigid part which helps in the erect position of the plant and to remain in standing position during the stormy time. Animals lack that cell wall because it does not require that much rigidity. Coming to the difference. Now animal cells are generally small in size whereas plant cells are larger than animal cell. Cell wall is absent in animal cell while a rigid cell wall made up of cellulose is present in plants. Plastids are absent except in case of protozoa and euglena. Plastid which help in giving coloration, chloroplast, chromoplast. That plastid is present in plant. Here vacuoles are many, small and temporary. Over in plants if we talk, permanent large central sap vacuoles are there. In animals, chromosome and centrioles are present. In plant, centriole and centrosomes are absent. This is a structure or the diagram of the plant cell and animal cell. If it is not clear to you, this diagram is not clear to you like this, you can go through your NCERT textbook. It is also having a good and a easy diagram. Whichever diagram you want to refer, you can refer, but this is one of the most important diagram for your class 9th. Conclusion. Now, if we talk about the conclusion regarding to the cell of plant and animal, so then both plant and animal cells contain membrane-bounded cell organelles like nucleus, endoplasmic reticulum, mitochondria, Golgi apparatus, peroxisomes, and lysosomes. Apart from that, both plant and animal also have similar membrane such as cytosol, cytoskeleton elements. Apart from that, plant cells can also be larger than animal cells, that is we know. The normal range of animal cells varies from 10 to 30 micrometer and that of plant is 10 to 100 micrometer. This was about our chapter. Here our chapter, that is the fundamental unit of life, gets completed. I hope whatever I have explained you through my PPT is clear. I did try to make it more easier for you all. But if it is somewhere something is lacking behind, then you please let me know in my comment box. This was me, Arpita Singh. I'll be back again with my next topic from next chapter. Till then, take care, stay home, stay safe. Thank you.